Yeah, okay. Hello, everyone. Um, I normally speak to a more general audience, so I am very excited to share this and not nervous at all about talking to a room full of Bromeliad experts. So I'll be talking about biogeography of Pitcairnia in the Caribbean. And we'll go through a little bit of background, focus on Dominican species, and talk a little bit about phylogenetics in the Caribbean, biogeography, and with conclusions and next steps. Okay, so for maybe the non-bromeliads or not the Pitcairnia people in the room, so Pitcairnia is one of the largest genuses, genera in the family. It is of neotropical distribution, and one African species is present in the group. And they're terrestrial, mostly growing on rocks, and they occupy a great variety of different habitats from high elevations, low elevations, humid forest, dry forest, you name it, they are there. So the reason why I'm interested in this group, particularly in the Caribbean, well, first I am Caribbean, so obviously there's a little soft spot for my species. But also Pitcairnia and Whitmackia show a very interesting patterns. They are very similar to each other. So together they represent most of the endemic bromeliad species in the Caribbean. Other groups obviously have endemics, but most of Whitmackia and all of Pitcairnia species here are endemic, and in both cases, most are island endemics. Also, you can see a similar pattern that Whitmackia in Jamaica and Pitcairnia in the island of Haiti or Hispaniola. There is an overrepresentation of species of these two groups respectively. So obviously the first thing I think of is, oh, it's the mountains, that it's driving all the speciation in these two islands, since these two are the most have the most and largest mountains of all the islands. Focusing more on Pitcairnia now, so we have 14 described species plus one identified as new but never published that we'll come back to in a minute. And we can see the pattern, mostly island endemics, the exception of Aditi and Pitcairnia angustifolia, which is present in a few of the islands. Okay, so before we go into the biogeography of this group, we need to clarify a few issues that are in the way of us getting there. The first thing is we need to understand better the relationship and identity of the species in Aditi. As you can see from here, all of these species were described a long time ago by Lyman Smith. However, these were all specimens collected by someone else and sent over to him. So he was looking at dry specimens, and in some cases, they are from cultivated specimens, so even not specimens growing in the wild. We can see up here, we have our undescribed species. I keep coming back to it because even though it's not described, if you go to the Smithsonian Herbarium and the herbarium in Santo Domingo, you will see specimens annotated with this identity because Robert Reed was very sure that this was a new species. He never described it. He died. I don't know how these events took place, but they are there identified as such. So we'll treat it as such for now. So when we look in the map of the very rough distribution of these species, we can see that they're kind of all over the place. So I've always thought that this was a bit strange, and we'll focus on Pitcairnia domingensis as an example. We can see from herbarium records that there are populations up here in the north, and then one outlier out here in the southwest of the Dominican Republic. if you are not familiar with the geography, these lines represent three very important mountain ranges, including the Cordillera Central, which is the largest mountain and tallest mountain in the Caribbean. And these two habitats could not be more different. So this being at the base of the mountain is a very dry habitat, and this is the rainiest part of the island. So a very, very humid area. So these two places could not be more different. So considering this, and this pattern repeats with some of the species, it always seemed odd to me that you would have, that you would have this distribution of very small populations, very localized. So you would assume that maybe the plants have a hard time getting around, but then they're like, this happens. Always seems kind of strange. Also, when we look at the characters that are used to differentiate between them, we see that the amount of scales, it's very important, like, and very striking. Like here you have a plant that's completely covered. Here it's not completely uncovered of scales. The presence of the spinal margins, it's also very different. That these don't appear to have any, and these are they're very hard. Trust me. So that combined with the lack of an identification key for the species in the Dominican Republic, if you ask anyone who has collected them there, they will tell you something like, oh, I have no idea which one I collected. Who could ever know? And when you go to the descriptions of these species, some of these characters are kind of overlapping in a lot of cases. So it is very hard to tell them apart. Okay, so we either have a problem with the species limitation or a problem with the species identification that we need to figure out. Okay, so I collected with my collaborators plants of Pitcairnia in most of their distribution. I completely ignored the name that was assigned to them in the populations in the herbarium, just trying to cover most of the distribution, which I'm very happy to report that we did, except for a couple of populations that I think are just gone now because of human activity. But nonetheless, we have a pretty good coverage of them. So we collected tissue for DNA extraction and herbarium records to later look at the morphology and to keep us records, obviously. So while I was waiting for more samples to put together my sequencing run, one of the professors in my department, Joe Walker, came to me and said, hey, I just found this paper that said you can extract RNA from silica-dried samples. I cannot believe this works. This is incredible. They immediately froze the tissue up to putting it in silica dried. How long has your Benad freeze? Oh, 3 to 6 months. Do you want to try it? Sure, let's try it. So we did, and we got RNA from our 3 to 6 months old silica dried tissue. I won't get into the details of that right now, but please look at our paper, read it. It's very fun. And so we got, even though this was a small representation, because we were just trying to see if it actually worked, but I included one sample from each location, and we're still able to get some very interesting patterns. So remember, there are no names assigned to it, just locations. And straight away, the first thing we see is that there is this first initial split here in the tree. And that split corresponds here in the top to the populations northeast of the Cordillera Central. And here at the bottom, the populations in the southwest of the Cordillera Central. These two specimens, 13 and 1, kind of like share some of the characteristics of both. You can see more of that in the paper. But more or less, it's a very, very striking division. So right away, we can see that the current delimitation, it's not correct. And so you might say, so okay, so what about the names of these species? So we asked Teodoro Clase from the Botanical Garden of Santo Domingo to identify them. So I gave him the specimens. I didn't tell them where they were from, just say, what name would you give them? And this is what he came out with. So you can see we highlight Pitcairnia elizabethae here. So you can see it all over the tree, as well as Pitcairnia domingensis. So we also did this exercise with the names that were in the herbarium specimens where we got the locations to go. And although they were different than what you see here in the tree, it's the same pattern. So nothing matches. There's no relationship between them. So right now we're pretty confident and thinking that these are just not the 5 or 6 species that we originally had. But what about the traits? Do you see any relationship of the traits with the tree? Again, no. So we have 3 of the traits here that we looked at, analyzed, and they're just there's no real pattern that you can see. So what we think is happening is that this might be a case for phenotypic plasticity. So we highlight this example, and I regret not having better pictures of it. But you can see here, this was by far the largest specimen that we saw and that we collected. It's a little bigger than me, although I know that's not saying much. But if you compare it to this one, it's gigantic. So these 2 specimens, we put them here on the tree. These are these 2. So genetically incredibly close to each other, more so than most other individuals between each other. And if we look at where they are in the map, they are, I don't know if you can read it, but 1.4 kilometers apart. So they're extremely, extremely close. This one is on a cliff directly facing the ocean with a lot of sun exposure, a lot of exposure to salt water. And this one, although, pretty close still to the coast, is really protected by a lot of vegetation. So not only do we think that the climate is very important, but the micro habitat of where these species or these individuals are growing, it's very important to how they look. So no wonder there were so many species identified. No wonder nobody could make any sense of it, because they're just very, very variable. So now that we know a little bit more about what's going on with the Dominican species, we need to know a little bit more about the Caribbean in general. Okay, so to the best of my knowledge, unless something has come up since, this is the only molecular phylogeny known for Pitcairnia or published. And this tree is a chloroplast tree. For this analysis, there was also nuclear markers used, but they did not resolve anything. So this is the only thing that we had kind of going for us. So here in this tree, we can see there are some issues, like some taxa that are not monophyletic, like Pitcairnia angustifolia, Pitcairnia integrifolia, and Pitcairnia domingensis, but we already know what's going on there. Also from here, we can see that all of the Caribbean species that were included are in this group. So at least we know maybe they're closely related to each other. But we see some transitions. One transition to the Caribbean and then others to Mexico and South America. Okay, so we need to have a better idea of the relationship between these species and the rest of the family without, going into doing the phylogeny for the entire family, which was not in my objectives. So we did sample, I have samples for most of the Caribbean and sampled outside our geographic area in Central and South America, concentrating mostly in Northern South America. And finally, we're getting to the DNA. So we did this with the Bromeliad 1776 kit, and using this pipeline to ultimately get an astral tree. So in this tree, I'll just highlight a couple of things. So we have here in blue this clade from Venezuela, Chulsei orchidifolia and venezuelana. In blue, Integrifolia, which is from Venezuela and Trinidad and Tobago, but this specimen is from Trinidad. And this species is actually very morphologically similar to the Caribbean one, so it makes also sense that this would be sister to this group. And then finally, we have in red the Caribbean clade. So I'm pretty confident that all the Caribbean species form a clade, and they are just the result of a single radiation into the Caribbean. So when we focus a little bit more on our group, we then see a couple of things. First, let me start here. So we have, again, our South America group also here. We're starting with Integrifolia to get into the Caribbean islands, or although is Trinidad and Tobago part of the Caribbean? It's a matter of opinion. Then we go here now fully into the Caribbean and only Caribbean species with Pitcairnia bifrons in the Lesser Antilles. And then we go to this group of Pitcairnia angustifolia, which is the one that was present in many islands. So this one, go back. So this one is in Puerto Rico and some of the lesser Antilles, like the British Virgin Islands and US Virgin Islands. I would like to highlight one species here, Pitcairnia yareki, which is identified and described for the British Virgin Islands. But from here, we can clearly see that it should be part of Angustifolia. Then we move on to Pitcairnia cubensis in Cuba. And then we go to our Dominican clade, which we recover, or Dominican and Haitian clade, which we recover the same groups that we did before. So here we have our northeastern clade and our south western clade, and this we can clearly see that we just had one single colonization event of the Caribbean, and then they just expanded and probably got isolated because, water and maybe mountains. So that's the pattern that we see here. So in conclusion, can say that species diversity is overestimated in the Caribbean. They do form a clade, so I'm very happy about that because it makes my life a little bit easier. And that colonization happened from North and South America, following the arc of the Lesser Antilles. So next, I still need to include some more samples to complete the story. I have some of these other ones I don't, and here I would like to ask all of you here, like if you can help me get some of these samples, and in some cases, some more of these samples. I would greatly appreciate it. I feel like there's no better group of people to ask this of. I will be performing like formal species delimitation analysis on the Dominican species. Right now, I'm kind of thinking I should include Angustifolia cubensis and Bromeliaceae here as well because they're just so morphologically similar. But that's to be determined. And we'll finally infer a dated and calibrated tree that we can do formal biogeography analysis for this clade. And finally, we'll get hopefully to an updated taxonomic review of this group and clear this mess a little bit. So Thank you very much to everybody that has been involved in this research. I don't know about raising a kid, but doing research really does take a village. So there were so many people that have contributed in so many ways. Feels kind of weird to say it's just like my dissertation, but we'll go with it. And thank you all. And I leave you with some pretty habitat pictures of Pitcairnia.
You for your presentation. Okay. Now we begin easily.
Absolutely fabulous talk. This was so exciting to see. I'm wondering if you could speculate a little bit about, the seed dispersal mechanisms in Pitcairnia are rather limited, right, compared to some of the other Bromeliad lineages in the Caribbean with plumose appendages, fleshy fruits, etc. How might the seed dispersal capability impact some of the biogeographic processes you're considering?
So yes, for sure. Some of these species, like you said, these are not like plumose species that fly so much around. They are more restricted in how far they can get along. I think that for sure is a barrier. However, we also do find them a lot around rivers. So, and like right growing, I don't think, yeah. So, like this one here, they just like throw their seeds into the water. So, I think that probably also helps a little bit with distribution. But I also hope to do some more population level analysis to see like how much flow there is from one place to another.
Hi, very nice talk. Really happy to see someone else doing something which is very similar morphologically with the Pitcairnias we have in. We are just laughing because they are so the same thing everywhere, and they are so diverse. This is crazy, I was just wondering because these angustifolia, do they have these leaf dimorphism with the spiky spines and the rhizome? Yeah.
Yes.
So then, how's the other ones?
In general?
I mean, all of them are like that.
So yes, and that's why I think I need to include all of those in the species delimitation analysis because, and I'm more familiar with angustifolia and the ones from the Dominican Republic because I went to the sites and collected them. And yeah, so you can see a variety of having a lot of spines. They usually, I think all of them like have in the base they have spines. I think that's constant for all of them, but around the leaves it is very variable. So yeah, in the bottom yeah, they were all very hard to collect.
One last question.
Great talk.
Thank you.
Couple points. Observation. You may not have caught my talk at the beginning of the conference, but we gave a pilot phylogeny for Pitcairnia using our Bromeliad 1812 kit. The results you found, the mixture of the names falling randomly, essentially, on your molecular phylogeny is just classic. And I guess I had two questions for you to clarify. First, when you presented the specimens to an expert, quote unquote, I guess I was wondering, A, what's the evidence he's an expert? And B, was there fertile material? And the second question is, how are these species differentiated from each other? Are they mainly vegetative characters, or are they floral characters or inflorescence characters?
Okay, so for your first question, he's not an expert on Bromeliad, so he is for sure. And and that's kind of why I was ignoring the names to begin with. For your second question, so it is the vegetative characters that are used to differentiate between them. So all the floral characters are basically the same, and like I said, the vegetative ones tend to overlap in the descriptions between each other. So
Thank you to the three presenters of this symposium.